Hello students, welcome back again. I am Samapti Sinha Mahapatra, your zoology teacher teaching in Sri Chaitanya Institute. Students, we are back with a new chapter today and we are going to discuss about body fluids and circulation. So before we start with the chapter, let's see what is the probable number of questions that we are expecting this year in NEET 2020. So since it is a part of physiology, this chapter is also important and frequently questions have been asked from this chapter. We are expecting 2 or 3 questions from this chapter this year in NEET 2020 as well. When you're talking about this body fluids and circulation, students, so definitely you're going to talk about some body fluid. So we have got different body fluids, but the main body fluid which we have got is blood and lymph. So we are going to talk about these 2 body fluids. Also, when you see what is the requirement of this blood, we studied the first chapter, digestion. The requirement was the end product of digestions like glucose, like amino acids, fatty acid, glycerols, everything which is ultimately utilized by these cells with the help of oxygen that we use, that we studied how it comes, how it diffuses in breathing and respiration chapter, which is going to help in the oxidation of food, but also how this oxygen is being transported. So for transportation you need a medium, and that medium is your body fluid. Mainly we are talking about blood. So let's see, in this flow chart we have given the division of the blood. Now looking at the parts of blood, students, this part is quite important for NEET. Questions have been asked in NEET 2018, as you can see I have written, and question also in NEET 2015 from this component, from this basic flowchart itself. So looking at the component of your blood, blood consists of plasma 55% and it consists of formed elements 45%. Now talking about the plasma, which is 55%, it has got different components. The major portion of your plasma is water. Can you see the percentage? It's 90 to 92%. So 90 to 92% of your plasma is itself water. And 6 to 8% is very important, some proteins. Now talking about these proteins, you have got fibrinogen and prothrombin, which is clotting proteins, because you will see in the clotting I will tell you that these 2 factors helps in the blood clotting. Talking about the largest size of proteins, that is albumin protein. These are largest in size and these proteins are responsible for maintaining the osmotic balance. These proteins, how they will maintain the osmotic balance, that we are going to see in the lymph part. Also talking about the third protein, which is globulin protein, which is of 3 types, alpha, beta and gamma. We will see that these globulin protein, alpha globulin, beta globulin helps in transport, whereas gamma globulins are responsible for production of antibodies, hence helping in the defense mechanisms. So different proteins, but important to know the function of each. Alright, then the remaining part is consisting of the different types of minerals like calcium, magnesium, chloride ions, etc. Also in your plasma you have got glucose, amino acids and everything in the transient state. So that's the composition of the plasma. Now talking about the formed elements, which we will be taking up in the next part in detail, it has got 2 parts. It has got blood cells, it has got blood platelets. You know very well the blood platelets are helping in the blood clotting, and blood cells are of 2 types, the RBCs, red blood cells, and the WBCs, which is the white blood cells. The white blood cell, students, gets further divided into granulocytes and agranulocytes. The one with the granules, the one with no granules. The granulocytes further divided into three types: neutrophils, eosinophils and basophils, whereas agranulocytes are further divided into monocytes and lymphocytes. So formed elements is 45%, this is the division, and plasma is 55%, and this is the division. Students also have written what is serum. If you want to see what is serum, serum is simply if you take the plasma and you remove the clotting factors, then plasma minus clotting factor becomes serum. Now students, let's talk about the different types of formed elements. So I have made a table and I have separated the different columns. The first column is the characteristics, then the RBCs, then the different types of white blood cells, which includes neutrophils, basophils, eosinophils, monocytes and lymphocytes. So we are going to do the comparative differences or similarities between these cells. So the first characteristics that I have written is the number. When you talk about the number of RBC students, that is way more higher than the white blood cells. As you can see, I have written it over here. The number of RBC is 5 to 5.5 million per mm cube of your blood, whereas the total number of white blood cells itself is only 6,000 to 8,000 per mm cube. Talking about the different percentages of different types of WBC, the neutrophils are 65%, which is maximum. The basophils are 0.5 to 1%, which is the lowest minimum. Talking about the eosinophils, it is 2 to 3%, and monocytes being 6 to 8%, lymphocytes being 20 to 25%. Very important to remember the percentage exactly because it is given in your NCERT. Alright, now talking about their shape, students. When you look at the shape of RBCs, it is biconcave. It is biconcave, it is not having any nucleus or cell organelles. And why it is not having nucleus? The reason you know, because you want to create more and more space for hemoglobin, and if you have more space for hemoglobin, hemoglobin is the one which is responsible for transportation of the gas, because even CO2 can bind with it, and maximum oxygen was transported in the bound state with hemoglobin as oxyhemoglobin. Now talking about the different shapes of the white blood cells. Now among the white blood cells, now you know that your neutrophils, basophils and eosinophils are granulocytes. So definitely they will have some granules. Whereas the monocytes and lymphocytes will not have any granules because they are agranulocytes. But the basic difference and the major difference between all is the shape of their nuclei. Talking about the very first, neutrophils, can you see this is the nucleus and these purple colored dots are the granules. The nucleus, as you can see, it is having not multiple nuclei, it is having multi-lobed nuclei. It has multi-lobed nuclei. Talking about the basophils, it is having S-shaped nuclei. Can you see S-shaped nuclei, or you can say bilobed nuclei, also having the granules. The eosinophil, students, is very well seen with a headphone shaped nuclei. So this is the headphone shaped nuclei, and this is also provided with some granules. Among all the white blood cells, the largest white blood cell is the monocyte. And the monocyte's nucleus, can you see the nucleus shape is horseshoe shape or kidney shape, or you can say it is bean shape. So horseshoe shape, kidney shape or bean shaped nuclei is seen in the monocyte, which is also the largest lymphocyte. Talking about lymphocytes, you can see that the nucleus is rounded without any granules. Students, there is an additional information you can remember, that when you talk about basophils, they take basic stain, as the name is suggesting. Eosinophil takes acidic stain, eosin. What about the neutrophils? Does it take neutral stain? Neutral stain is nothing, obviously. So the neutrophils are responsible, or they take both acidic as well as basic stain. Got it? Now talking about the site of formation. The RBCs during the embryonic stage is also erythropoiesis we are talking about, right? When you say erythrocytes, definitely you are saying erythropoiesis, formation of RBC. When you are saying white blood cells, or you say it leukocytes, so definitely formation is called as leukopoiesis. Right? Erythropoiesis starts in the yolk sac in the embryonic stage, and in the liver, and in the bone marrow. So multiple sites of formation of RBC. Talking about the white blood cells, bone marrow is common for all. Neutrophils bone marrow, the basophils bone marrow, eosinophils bone marrow, monocytes also bone marrow. But talking about lymphocytes, they are not only formed in the bone marrow, they can be formed in other lymphoid organs like in the tonsils, like in the thymus, or in the lymph nodes. That's what I have mentioned. That's the site of formation. Talking about the lifespan, you people are very much familiar. I have not made a column. You are familiar with the lifespan of RBC, which is 120 days. The lifespan of RBC is 120 days. Whereas when you talk about white blood cell students, the lifespan varies, and varies in the blood, varies in the tissue, but they are very, very short-lived. But the lifespan of RBC is 120 days. Now, what is the function? We now very well know, since we have done breathing and respiration, that the function of your RBC is definitely transport of gases. So transport of gases with the help of hemoglobin, because the oxygen is binding with it, right? Talking about the white blood cells separately. Remember that the neutrophils among the granulocytes, the neutrophils are the phagocytic cells. It performs phagocytosis, engulfing everything, alright? And the neutrophils also, students, are the first cells to come and appear at the site of injury. Very first cells appearing at the site of injury is the neutrophils. Talking about the second one, basophil students, it is going to produce heparin, histamine, serotonin. Do you remember any other cells which produces heparin, histamine, serotonin? And do you know when it is produced? So definitely they act like your mast cells. You know that mast cells also produces serotonin, heparin and histamine, and they are involved in the inflammatory reactions just like the basophils. So we can say that basophils acts like your mast cells. You know the function of each. Heparin is anticoagulant, histamine vasodilator. This is the blood vessel. When histamine acts, vasodilator, blood vessels dilate. When you talk about serotonin, this is the blood vessel, and serotonin acting blood vessels constriction. Talking about eosinophils, it is produced mainly during the allergic reaction. Remember that. Now, among the granulocytes, the neutrophil was phagocytic in nature. But talking about the agranulocytes, monocytes and lymphocytes, your monocytes are phagocytic in nature, and it is also called as the macrophages of your blood. You know that different macrophages had different name. The macrophage of lungs is called as dust cells. The macrophage of liver is called as Kupper's cells. The macrophages of your nervous tissue is called as microglial cells. The macrophages of your blood is called as monocytes. The functions of lymphocyte students, you know that in your human health and diseases, you must have studied that you have got two types of lymphocytes. One is T and other is B. And both the lymphocytes are associated with some types of immunity, the cell-mediated immunity and the humoral immunity. So overall function of the lymphocyte is to provide you with immunity. So that's the comparison. You're going to remember the data at least as it is. Also, I would want to tell you a specific term that we use for the white blood cells, and that term that we use for the white blood cell students, that term is called as diapedasis. The term is diapedasis. What do you mean by this diapedasis? Diapedasis is related with the movement of white blood cells. The white blood cell, students, for its functioning, it can change its shape. Suppose this is the white blood cell, it is always going to squeeze like this, squeeze out of the blood capillaries, and this changing in shape and squeezing out of the blood capillaries, this movement of the white blood cells is called as diapedasis. Next we are going to discuss about the function of your blood platelets. Now students, let's talk about the functions of the blood platelets and its different characteristics. Talking about the blood platelets, they are also called as thrombocytes. Alright, they are also called as thrombocytes. From which cells they are formed? They are formed from specialized cells called as megakaryocytes. Formed from megakaryocytes. They are basically, students, the cellular fragments. They are basically the cellular fragments, not the actual cells. Talking about their number, they are 1.5 to 3.5 lakhs per mm cube of your blood. Their lifespan is approximately 1 week. Definitely the function I have written helps in blood clotting. Students, blood platelet is very important because the blood platelets, they produce several factors that helps in your clotting of the blood. If in any way your number of blood platelet is less, blood platelet count is less, then you are going to suffer from excessive blood loss. Now since we are talking about the functions of the blood platelets, let's start with first the process of blood clotting. How this clotting is actually taking place. We need to know which is the first factor or which is the first condition that is required for blood clotting. Can you answer me? Actually, the first thing that you require for blood clotting is injury, right? So whenever there would be an injury, that's what I have written. Whenever there would be an injury, that is going to stimulate the blood platelets. And injury, when it is stimulating your blood platelets, this is what I have written over here. Your blood platelets start producing thromboplastin. Students, here important to note which factor is produced first, then second, then third, then fourth. Okay, write that. So the first thing that is produced is thromboplastin, and this thromboplastin I have written in bracket is also produced by tissue at the site of injury. The tissue also start producing factors called as tissue thromboplastin. Clear? Now this thromboplastin via cascade of reaction, I have written it over here, that thromboplastin ultimately forms an enzyme complex. It forms an enzyme complex called as thrombokinase. And this thrombokinase is not formed directly from the thromboplastin, students. It is formed as a result of cascade of reaction, series of reaction. Once your thrombokinase is formed, it is responsible for conversion of prothrombin inactive into thrombin which is active in presence of calcium ions. So now you know why calcium ion is also very important for blood clotting. This thrombin, upon activation, is converting this inactive fibrinogen. You remember prothrombin fibrinogen was present in your blood, in your plasma, helping in clotting. That's how they are helping in clotting. So this fibrinogen, which is soluble in the plasma, right? Which is soluble in the plasma, is getting converted into the insoluble, the network of threads, the clot that you actually say, the brown color thing that you say, called as fibrin in presence of again calcium ion. So only remember the sequence: injury stimulating blood platelets producing thromboplastin, thromboplastin forming thrombokinase, thrombokinase converting prothrombin into thrombin in presence of calcium ion, and this thrombin converting fibrinogen into fibrin in presence of calcium ion. That's how clotting is performed. Now students, let's start with a very easy topic. Since junior classes we are studying this topic, which is also very important for all of us to know, that is blood grouping. We all should know what's our blood group, who are the donors, who are the recipients, and so on. Now talking about the blood grouping, we have two types of blood grouping. One is called as ABO blood grouping and the other is called as RH blood grouping. Talking about ABO blood grouping, I have shown you in tabular form that what could be the different types of blood groups. What if the person is having A blood group? What does that mean? What is the antigen over RBC? What will be the antibodies found in the plasma? Who is the donor? Who is the recipient? Now students, remember one thing. If I say the person is having X blood group, remember the antigen on the surface of RBC will be X. If I am saying person is having A blood group, that means the antigen on the surface of RBC of such person will be A. If I am saying the B blood group, then the antigen on the surface of RBC of that person is B. If I am saying AB blood group, then both A antigen and B antigen is present on the surface of the RBC. If I am saying O blood group, O means the person is having no antigen on the surface of RBC. Is it clear? Now definitely students, whatever is my blood group, I am never going to form antibodies against it. I will be forming antibodies against all other blood group which I don't have. So suppose I have A blood group, I had A antigen. Now these antigens, students, don't get confused between the foreign and this and that. These antigens are the self antigens. These are the glycoproteins present over your RBC. These are not the foreign particles. But if the person is having A antigen on its surface, the antibodies that it will have will be against B, and antibodies are written as anti-B. So anti-B means antibodies against B. If I am saying B blood group, antibodies against A. If I am saying AB blood group, since it has both the self antigen A and B, it is going to having nil, no antibodies. If I am saying O blood group, since it had no antigen on its surface, it will be having antibodies against both A as well as B. Got it? Now we are going to see who will be the donor if blood group is A, B, AB and O. If your blood group is A, so who can be your donor? Now if your blood group is A, now you know that you have antibodies against B. So definitely B will never be your donor. In fact, AB blood group cannot be your donor because it is also having surface antigen B. So definitely your donors will become A and O, because this person having A blood group is not having any antibodies against A and any antibodies against O. In the same way, if your blood group is B, your donor can be either B or your donor can be O, because person with the B blood group has no antibodies against B and against O. But if you donate A to the B blood group person, it is having antibodies as antibody against A. Clear? If you have AB blood group, do you have any antibodies? You don't have any antibodies. You can receive blood from any person, A, B, AB and O. And that's the reason why AB blood group is universal donor. Actually, you will say that AB positive, I will tell you why. AB positive is universal donor. Talking about the last blood group, which is O, this is universal recipient, and specifically O negative is universal recipient. I will say why, but O can receive blood only from O because it is having antibodies against both A as well as B. So can only receive blood from O blood group person. Now talking about positive and negative, since I mentioned, it is about the RH blood grouping. Now what is this RH? This RH is coming from the rhesus monkey, which also has similar kind of glycoprotein. There are only two conditions. Most of the persons are RH positive and some are RH negative. Now if I say the person is RH positive, remember that it is having RH antigen over the RBC. If a person is RH negative, it is having no RH antigen on its surface. Now, why did I say that only AB positive is a universal recipient, not the negative? Because positive can receive blood from positive as well as negative, but negative cannot receive blood from positive. Negative, since it cannot receive blood from positive, because positive means having the antigens, it will form antibodies. Hence, AB positive is a universal recipient. But when you talk about donating blood, O negative is a universal recipient, not the positive, because negative person can receive blood from negative as well as it can, negative person can donate blood to negative as well as it can donate blood to positive. Because negative means having no surface antigen, so none of the person will form antibodies against it. So negative can give blood to negative as well as positive, hence O negative is a universal donor. So that's a very easy concept. You will be asked direct questions about donor and recipient and so on. Now, talking about a condition that is related with the blood grouping that we discussed just now is called as erythroblastosis fetalis. It is called as erythroblastosis fetalis. Now, students, remember the conditions. Only in those conditions erythroblastosis fetalis is going to take place. So, I have made a flow chart which is going to make it easier for all of you to understand. Now see, the condition, very first condition of erythroblastosis fetalis is that the mother should be of negative blood group. What happens is mother should be of negative blood group, and if she is pregnant for the first time, during the first pregnancy, the fetus, the baby that she carries is having what blood group? Positive blood group. So listen carefully, the mother should be negative blood group, and during the first pregnancy, the fetus should be positive blood group. Now when you say positive blood group, that means the fetus is having antigen Rh on its surface, whereas mother is not having any Rh antigen. But during the first pregnancy, there is no problem because the mother's placenta is acting as a barrier. So there is no problem during the first pregnancy. What happens after delivery? The mother is coming in contact with the negative blood group. When the mother comes in contact with the negative blood group, in the positive blood group of the fetus, what happens is that mother starts developing antibodies, because mother was having the negative blood group but the fetus is having positive blood group. The mother starts forming antibodies against that positive Rh antigen as soon as after delivery she comes in contact with that Rh positive. So that's what I have written, that mother in contact with the fetus starts forming anti-Rh, and when I say anti-Rh, that means mother is having antibodies against Rh. Now what will happen in the next part during the second pregnancy that we are going to see. Now when the mother this time is pregnant for the second time, again mother is having the same negative blood group, but this time what she has is anti-Rh, which means antibodies against Rh. And when she is pregnant for the second time, the fetus again that she is carrying is having Rh positive blood group. Now what is going to happen? In this case, the mother which has developed antibodies against Rh, anti-Rh antibodies, that is going to cross the placenta, and that is going to cause the lysis of the RBC of the fetus, which is why this disorder is called as hemolytic disease of the newborn. What you are doing? Hemolysis, breakdown of the RBC. But the question is, why in the first case I am saying placenta is acting as a barrier and nothing is happening, and in the second case I am saying that the antibodies are crossing placenta? Is it necessary, students, that the blood group of the mother and fetus is same? Mother might have A blood group, fetus might have B blood group. Why not in that case the antibodies against B is crossing the placenta, and only these antibodies are crossing placenta? The reason behind that, the antibodies which are these are very small in size. They are IgG type. So being very small in size, they are able to cross the placenta easily. They are going to cause the lysis of the RBC. The fetus generally develops jaundice. The baby generally develops jaundice and anemia and may die. That is called as erythroblastosis fetalis or hemolytic disease of the newborn. But it can be prevented. How it can be prevented? If after the first delivery, when the mother came in contact with the Rh positive, what we can do is provide the mother with anti-Rh, antibodies against Rh. All those antigens will then be destroyed and we will not allow the mother herself to generate antibodies. If the mother is not generating antibodies, we won't have any antibodies during the second pregnancy. That's how we can prevent erythroblastosis fetalis. Now students, the next body fluid that we will be discussing is the lymph, which in NCERT is written in bracket that it is also called as tissue fluid. First of all, let us see how this tissue fluid is formed. As the name itself is suggesting, tissue fluid, so it is a fluid actually which is present around the tissues. Now in this diagram I have made, this is the artery, this is the vein, and in between you have capillaries. The arrangement is like that only. Arteries forming thin network called as capillaries, merging to form your veins. Now the blood is coming through your artery, is passing through the very fine capillary which is lined by the endothelial cells, and it is getting filtered out. Once it is getting filtered out, it is getting accumulated around the tissue, forming the tissue fluid, forming the lymph. But students, there are some part of blood or plasma which is not filtered out. The part of the blood which is actually not filtered out is RBC. There is no RBC, which is why your lymph is colorless. You don't have those proteins which maintains osmotic balance, that is albumin, because that is being very large in size. All other factors are present in your lymph. But the lymph is not only accumulated around the tissue space, lymph is actually acting as a middleman. I have written over here that lymph acts as a middleman. When you say middleman, students, can you see it is present in between your blood and tissues accumulated around here. All the nutrients, gases and everything is going to reach to the tissues and come back only via this tissue fluid. So acting as a middleman. But this tissue fluid, as we are moving our skeletal muscles, as we are doing more movements, it is moving into some other part. That's where I am talking about the pathway of lymph, or you can say lymphatic system. What is happening? Your lymph is formed. Lymph is then carried out by the network of capillaries called as lymphatic capillary. From the lymphatic capillary, student, which is only having one open end, this is also called as blind end. From the lymphatic capillaries, it is reaching into the larger vessels called as lymphatic vessels. We have got lymph in our right part as well as left part. So on the right side, the lymphatic vessel is called as right lymphatic duct, whereas on the left side it is called as left thoracic duct. From these lymphatic vessels, lymph will ultimately mix into your blood on the right side in the right subclavian vein, on the left side in the left subclavian vein. But before you send this lymph into the respective veins, into the blood, it is having different antigens as well, because it was in contact with the tissues. So you need to filter it out. Before you send the blood from the right lymphatic vessel into the right subclavian vein, and from the left thoracic duct into the left subclavian vein, you filter it using the lymph nodes which are present at several sites in your body. Lymph nodes has lymphocytes which is going to filter out this antigens, remove everything, and and then it is ready to be sent into the veins, which ultimately merges with the superior vena cava, which ultimately sends blood into your heart. So that's the pathway. Although pathways are not that much asked frequently, but it's very easy to remember. So just follow this step and know the composition of the lymph. That's how you complete your two main body fluids, your blood and your lymph. Now students, let's start with the next part of the chapter, which is the different circulatory pathways, the different types of circulation, the different types of heart. Now talking about the general information about all the vertebrates or invertebrates, the types of circulatory system that we can have is open circulatory system and closed circulatory system. Now whenever we say that anyone has got open circulatory system, that means the blood is going to flow through the cavities or the sinuses. There is no network of closed blood vessels. So this open circulation is seen very well in the arthropods or the cockroach. Very common example. But when you are saying closed circulatory system, that means for the blood to pass through, there are network of blood vessels, arteries, veins and everything through which the blood is pumping. Very common example, we know that humans also have got closed circulatory system, amphibians, fish, reptiles, closed circulatory system. So that's about these two. Now talking about the types of circulation, animals may have single circulation, double circulation or incomplete double circulation and mixed circulation. Now talking about the very first, single circulation. As the name is suggesting, single circulation. So the blood in case of single circulation is pumped only once through the heart before reaching to the tissues. It is very well seen in the fish. Now I've written one more information about the fish. That fish has got two-chambered heart. This two-chambered heart means it has got one atrium and one ventricle. But the fish additionally also has a chamber called as sinus venosus. That's the additional chamber. Now talking about single circulation, actually the heart of fish is also called as venous heart, because the fish is pumping, heart is pumping deoxygenated blood. It reaches to the gills for oxygenation. Once it reaches to the gills, all the oxygen is gathered, blood becomes oxygenated, that blood is transported to the tissues. So it is not again coming back to your heart and then to the organs. So blood is pumping only once to the heart, going once through the heart. Heart is pumping to gills, gills is giving to tissues. That is why this type of circulation is single circulation. Now talking about the mixed circulation or incomplete double circulation, seen in amphibians and reptiles, which has got three-chambered heart. Now the exception in reptiles is definitely crocodile, but talking about a general property, amphibians and the reptiles, they have got three-chambered heart. They have got two atria but a common ventricle. Since they have got a common ventricle, definitely on the right side deoxygenated blood, on the left side oxygenated blood, but both is getting mixed in the common ventricle. And since it is getting mixed in the common ventricle, it is pumping the mixed blood, hence it is called as mixed circulation or incomplete double circulation. There is one more information I have written about the amphibians. Just like the fish has got definitely two-chambered heart and additional chamber sinus venosus, amphibians also have got two additional chamber, one exactly like that of fish, which is sinus venosus, and the other is called as truncus arteriosus. So that's an additional information. Talking about the last type of circulation, double circulation. Double circulation we are going to study in detail because humans have got double circulation, or we say that birds and mammals which have got four-chambered heart, the atria and ventricles are completely separated. We have got two atria, we have got two ventricles, and blood is separately flowing deoxygenated through the right side of the heart and oxygenated through the left side of the heart. There is no mixing called as double circulation, which we are going to discuss in detail. So that's about a general idea about circulatory pathways and the type of circulation. Now students, in the next class we are going to start with the structure of human heart or the type of circulation in the humans. So before that, go through this entire part from the NCERT as well. We are going to come back soon. We are going to start with the structure of human heart in our next class. Till then, take care, stay happy and bye bye.